Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in creating a better tomorrow. Today, we have the honor of being joined by Dr. Jay Hooper, who is the chief of the Molecular Virology branch at the United States Army Medical Research Institute of Infectious Diseases, also known as USAMRIID, where his research is aimed at the discovery and the development of medical countermeasures targeting various exotic viral diseases of military importance, including hemorrhagic fever diseases caused by hantaviruses, arenaviruses, filoviruses, which include Ebola, as well as various diseases caused by pox viruses, including smallpox and monkeypox. Dr. Hooper has made numerous discoveries in the domain over the years, including one of the world's most potent monoclonal antibodies targeting smallpox, one of the first practical lethal disease animal models for hantavirus, as well as multiple hantavirus DNA vaccines that have moved into phase 1 and phase 2 clinical trials, as well as some rapid development candidate antibody products and vaccines for Ebola. During COVID-19, Dr. Hooper led an effort to establish the rodent animal model to discover and advance medical countermeasures targeting COVID. And that work resulted in one of the first publications of the Syrian hamster model, as well as the human ACE2 mouse model to evaluate these countermeasures. And then those were used extensively in the developing of vaccines and antibody treatments during Operation Warp Speed. Dr. Hooper is currently collaborating with various industrial partners on future generation vaccines and antibodies in the area of monkeypox. He did his PhD in virology at Harvard, undergraduate in biology from Colby College, and we're honored to have him with us on the show today. A lot of cool topics to get into. Dr. Jay Hooper, welcome to our show.
Yeah, thank you for having me. Thank you for that introduction.
It's an awesome
Reminded me of
introduction.
a lot of things.
No, you sure have been up to a lot of stuff. A lot of really cool topics to get into. I'd love to start off, Jay, sort of, at the beginning to learn a little bit more about you. Just a little bit more about your background. Obviously, your segue from biology into virology, and I found it quite interesting. I took a look, as I typically do, at your dissertation, which was on mechanisms of something known as reovirus entry into cells, and I had to do a little research on that. And interestingly, we in that class, falls rotavirus. We had Paul Offit on a few months ago talking about all his work on the vaccine, but it's a very interesting area in the sense that a lot of those viruses actually aren't too pathogenic, like some of the nasty stuff you deal with now, and in some cases, like have interesting oncolytic properties and all that for drug development. Talk a little bit about your PhD, if you would, and a little bit of how that class of viruses sort of fell into your lap at the beginning.
Yeah, so in grad school, I ended up in Bernie Field's lab. People might recognize the Field's name from Field Virology. Anyways, what his thing was reovirus, and he was one of the first people to start mapping phenotypes to genes of a virus. And that was possible because back in those days, you could make reassortant viruses. So you could mix a couple reoviruses with different phenotypes together, and so you could map the phenotype that is responsible for that different property that virus has. So it's kind of like we called it Stone Age virology. And so I went there and I was interested in trying to figure out how a non-enveloped virus, so reoviruses don't have the fatty envelope that a lot of viruses like SARS-2 have, and almost every other virus that we work with, other than polio, has an envelope, but reoviruses don't. So how does that particle interact with access to host cells? So that was the question. And myself and others were able to map that to a particular gene. And then we made different mutants that had different, slightly different properties in how their stability and how they penetrated the membrane. And so we were able to map in what part of the protein was responsible. And then I used some monoclonal antibodies that existed to see if those could inhibit the property. And I was measuring cell entry, but using a chromium release assay, which nobody really does anymore. As chromium 51 cells are loaded with that, and when their membrane is perturbed, they release chromium, and you measure that in the media. So that was kind of the big assay that we were using. And so we were able to make some progress, and it was interesting. Right about that time, there was a lot of cryo-EM going on and getting the structures of the different forms of reovirus. Steve Harrison at Harvard was doing a lot of that, getting crystal structure of these proteins. And so as we were kind of doing the Stone Age of virology, the structural biologists were starting to show us what it actually looked like. And we could kind of start to see all of the things that we were observing in cell culture and those kinds of things were now being mapped onto structures, and could start to see kind of what was going on. I left before all of that was solved, but it was an interesting problem, and that's what I was doing there. But I was interested in more. That was really, really academic, and I was more interested in doing something that had really applied utility. It's kind of interested in vaccines and other medical countermeasures to prevent viruses from being the problem that they can be. And so that's really where I steered myself to go get into that. And USAMRIID turns out is a great place to look at those things.
Yeah, it's a very interesting hop from, again, not that the reoviruses aren't that bad, but to where you went next, and the type of stuff that you've been doing the last couple decades. Really sort of a major leap. Jake, could you talk a little bit about USAMRIID? I mean, we've talked to, other folks on the show that we profiled from other groups that fall sort of, I know a lot of these organizations that do this and other sort of CBRN research fall under the United States Army Medical Research and Development Command, groups like Walter Reed Army Institute, the United States Army Medical Material Agency. USAMRIID is another one of those sort of subordinate agencies, but you are the only DoD lab specifically focused on biosafety level 4 type organisms. Introduce us to USAMRIID, if you have. I guess we hear a lot about it in the movies, but talk a little bit about where you are, and then a little bit of I guess what's like getting a job there. I mean, it's not. I guess you didn't apply for it like a typical, hey, I want to study vaccines, but talk a little bit about that.
Yeah, so USAMRIID is the U.S. Army Medical Research Institute for Infectious Disease. It's in Frederick, Maryland. Before USAMRIID existed back in let's say the 50s, it was Fort Detrick, and it was an offensive and defensive biological, weapons research lab. And then when the U.S. signed the biological, the treaty to end all offensive biological weapons research, that's right around the same time that the actual USAMRIID was built and set up. That was in, I think, 1969. And so we're still in that building. has a high labs, BSL 2, 3, 4. So 4 is the one of the highest, basically the suit research. And so the Army is interested in biodefense, but also in defending against natural threats that pose a risk to the warfighter or the military. For example, hantaviruses are viruses that the military can run into out in the field, carried by rodents. During the Korean conflict, there was about 3,000 cases of hemorrhagic fever with renal syndrome caused by hantaviruses. So diseases like that require high containment, we also work on those. And right now, the term emerging infectious disease, emerging pathogens has become, more and more popular, I guess. These are the zoonotic diseases that are kind of rare, but they come out and can really cause problems. And those, the military has run into those out in the field for years, but now numbers are increasing for certain of those diseases. And so, it's becoming a more widespread target of NIH research. And so we do a lot that's very similar to what the high containment labs for NIH would be doing.
Mm-hmm. Mm-hmm. And along those lines, because clearly, there's not a lot of these BSL for labs in existence. These are skills that one picks up, I guess, over time in, beginning to work with these, pretty nasty pathogens. And I joked around, a couple months ago, I profiled Colonel Tina Schoenberg over at USANRIID, and she was talking about sort of this functional Area 52 specialist role in her organization in Fort Belvoir, these nuclear technicians that got to deal with, cleaning up nasty stuff on the nuclear front. But any interesting stories, anecdotes related to sort of the requirements and what you went through and becoming sort of good at the BSL-4 processes, because clearly at some point we say, hey, Jay, they've got some weird looking stuff over here we want you to analyze and, put on the suit and all that. You got to be good at it. You can't mess that stuff up just playing around. So talk if there's anything on that front, because I think that's an important piece of sort of the development of your skill set.
Yeah, I guess for me, I was doing a lot of work at BSL-3 for years before ever going BSL-4. And so BSL-3, there's different postures you can have at BSL-3. You can go up to using PAPRs, which are purified breathing apparatuses, that kind of thing, even at BSL-3. And you have to really be very careful at BSL-3, because the way we used to do it at USAMRIID is we did not have really enhanced BSL-3. So basically you were in scrubs and you were in a hood. And so you had to really know what you were doing. These were not the BSL-4 agents that we were working with, but you get good at being safe. Now, when you go to BSL-4, a lot of us joke that BSL-4 is, easier than BSL-3 because you are in a spacesuit. There's more wiggle room as far as I think really safety, because of you're doing all the work in hoods, but you also have a suit on. So you're not donning and doffing with the same kind of risk that you are when you're in BSL-3. I don't know if that really makes any sense, but the big thing about BSL-4 is they make sure that you're, not claustrophobic. You don't easily kind of panic if certain things happen, because you are in a situation where you can't panic. So as far as kind of anecdotes go, the one that's actually, it's not really funny, but it is kind of funny. I had a technician who was in BSL-4 in a blue suit, So that's kind of the older version of the spacesuits. We call them the blue suits. Now there's white suits. It's also orange suits, but he was in a blue suit, and it turns out that the air source that supplies air. I don't know exactly the engineering how it works, but there's pumps that basically continually remove condensate from the kind of the air intake areas, but those must have went down. So he was still getting air, but so he's in the blue suit, and not only is he getting air, he's also getting water. He said it was very rank. It was kind of mildew, kind of the kind of water that would be sitting in
Mm,
something before being pumped out of a
mm.
kind of an air conditioner type unit. And it was pouring in on his head and basically starting to fill up the suit, down around his feet and working its way up. And so that's the kind of time that you don't want to panic. So he was
Right.
able to slosh his way to the shower. It's a kind of a disinfectant shower to get out of there. But um, he did get out. Everything was okay. But that's a situation where you you might want to panic. But
Right.
he didn't. He was fine. But kind of a shocking thing that happened there.
Yeah.
And I've only heard of that happening once in the 30 years that I've been here. So
It makes again makes for some good cocktail hour trivia, no doubt. But
yeah.
no, I appreciate that. Um, Jay, as I mentioned in the bio of the show, one of the things that you're actively involved right now is collaborating in sort of the vaccine and antibody area related to everything going on with monkeypox. But it's interesting that your monkeypox research started a while ago. You had a really interesting paper about 20 years back in the Journal of Virology in 2004 May, smallpox DNA vaccine protecting non-human primates against lethal monkeypox. And then a couple years later, around 2011, you wrote this interesting piece on the strategic use of smallpox vaccines in the so-called post-eradication world. And, here we have a disease, and I know it's always been debated, like, hey, we've eradicated this thing, why do we keep it around? But hell, as you point out, there are other pox viruses, other orthopox diseases, like monkeypox and other exotic ones we might not hear as much about. I was wondering if you could just talk a little bit about sort of the early monkeypox research you were involved in, and then a little bit about sort of how this discussion debate around sort of getting rid of this stuff from the planet, um, you know, what goes on with this debate in 2024? Because clearly we have a use case, unfortunately, going on right now where, we need sort of the smallpox proxy to deal with this.
Yeah. So yeah, way back, I was involved. Actually, I was a technician at the time. I should mention I was at USAMRIID before going to grad school as a technician.
Neat.
So yeah, you asked how I got kind of moved from biology to virology. I was a bio major at Colby, trying to find a job. I'm from Maine, really no jobs up there at the time. No connections in Boston, where where I needed connections. And it was kind of it's a long story. I'm not going to go into it, but I bumped into some recruiters who were trying to get people into a program called the O&H program, which was for graduate, college graduates. You'd go in the army, you'd do 3 years in a lab.
Mhm.
Um, and so I talked with a scientist down at USAMRIID, and the research sounded really interesting. And so I came down. Did 3 years. And during that time, the project I was involved in was isolating monoclonal antibodies to vaccinia.
Okay.
So vaccinia is a pox virus. It's
Yep.
kind of a BSL-2 virus, but we were isolating monoclonal antibodies, and we isolated a lot. And we were looking at their properties like neutralization and that kind of thing. And we found a very, very potent one that turns out to be that 7D11 monoclonal antibody. But we found other monoclonal antibodies that also protected. for example, against the A33 protein. And so all that was kind of going on where we were starting to identify genes of pox viruses that contributed to protection. And we knew that from the monoclonal antibody work. So when I came back to USAMRIID, 6 years later, started doing DNA vaccine work. And first thing I did was take some of those targets of those monoclonal antibodies, vaccinate mice with those, and see if we could get protection. And what we found was we could, especially when we combined, multiple genes targeting the different infectious forms of a pox virus, because there are a couple different infectious forms. And so we ended up settling on a combination of 4 genes, 2 to each type of particle. And we called it 4-pox. And we did that study that you referred to in non-human primates. That was actually the first time monkeypox had been in NHPs for this kind of testing a medical countermeasure since the 60s. It's been a long time, and John Huggins was doing some work on kind of bringing that model back online. And so the model was we're dusting it off. And so we tested the DNA vaccine in that model, and it protected against monkeypox. So that was pretty exciting. That's that paper that you talked about. And then we continued. We looked at actually other platforms like alphavirus, replicon particles. So that's like a virus vectored vaccine platform. This combination of 4 genes worked in that. We worked with Veffa Francini at NIH to do a protein version of 4-pox that worked. We did that study kind of comparing head-to-head against MVA, and that's where we started to see, how good this DNA vaccine was. So, and we were delivering the DNA vaccine by various technologies, ranging from gene gun to electroporation. And right now we're using jet injection. But when the monkeypox, we always kind of knew that monkeypox was kind of smoldering over there in Africa. And it was starting to pick up steam a little bit as the population who was originally vaccinated against smallpox, as they kind of aged out and the population became more and more naive, you're starting to see more and more monkeypox because the smallpox vaccine protects against monkeypox.
Right.
And so then that kind of crazy outbreak that started in around 22 happened. And my problem with DNA vaccines had always been and remains the difficulty in getting an industrial partner to really kind of take it the distance.
Got it.
And so, after COVID, seeing how the messenger RNA platforms had really kind of exploded as far as kind of their industrial sophistication, getting those vaccines out. I talked to Mrna, and they were interested in monkeypox, and so we went forward, and it turns out it's the same targets. The monkeypox ortholog versions of them is the current vaccine that Mrna is taken into a phase 1 clinical trial at this point.
Very cool.
But yeah, back to that review. What we were kind of concerned about is the ACAM 2000, which is the vaccine, the smallpox vaccine that we know works because it is essentially identical to Dryvax and other vaccines that were used during the smallpox eradication. So it's a very effective vaccine, but it does have safety issues. So, vaccines all have risk, but it kind of depends on the risk benefit. How big a danger is there that you need to have a vaccine for will kind of dictate the amount of risk you're willing to put up with. And there's so many issues with ACAM 2000 as far as safety goes that it's not really routinely used. And so that kind of leaves us a little bit vulnerable. So we were interested in figuring out strategies, whether they be a new vaccine that's safer or ways to use molecular vaccines to try and give the population more protection against a possible outbreak of something like smallpox, which would really be a problem. So that's what that review is kind of discussing, ways of using molecular vaccines to boost people with waning immunity, to prime, that kind of thing. Yeah, I forgot what year you said that was. Was that
That was 2011.
10 years ago? Yeah. So, yeah, I guess, we didn't predict that the zoonotic would turn into a pandemic or a large kind of worldwide outbreak like it did in 22. We didn't see that coming, but I think that's the thing with zoonotic diseases, like we saw with COVID also, is that you never know what's going to happen when they get in the population. Things evolve, things happen. So here we are, 2024.
Yeah.
Yeah.
And yeah, I mean, speaking of things getting to the population and the unknown, clearly you were quite busy at USAMRIID. As mentioned, the Syrian hamster model, the ACE2 mouse model. Obviously, these were key in sort of both the vaccine and the antibody side of stuff. Take a little time to tell us what you've been up to sort of between 2000 and Monkeypox, and everything Operation Warp Speed in terms of where you were, sort of what types of marching orders you were getting at the time. Clearly, I'm sure there was a lot of stakeholders across the entire sort of anti-infective system of the Department of Defense that were contacting you about this stuff. But tell us a little bit of that story, if you would.
Yeah, I'll just say, like, during that time, I've been hammering away at the hantavirus problem, trying to get a vaccine. But also during that time, we kind of would have outbreak after outbreak after outbreak. And so, back in 2014, kind of Ebola kind of blew up on the scene. My role in that is I have a team in my group that has developed pseudovirion. So they're basically fake viruses that against almost every virus that we work with, except for pox. We can't do it for pox. But we did do it for all the different variants of Ebola. And so what we were able to do was, there were multiple different Ebola vaccines that the government was kind of helping to move forward toward the clinic and beyond. And we ended up doing the neutralization assays for all of those. And it was really interesting because we were able to, as the US government, we were able to compare in the same assay, the neut responses to the different vaccines. We were able to compare the human responses to the non-human primate responses, all in the same assay. So we had really good visibility of what vaccines look good, not so good. So that was pretty exciting. And one of those vaccines, was the VSV-based Ebola vaccine that ended up being licensed by Merck eventually. So then, after that, kind of Zika pops up. We had a Zika nude assay going up. We quickly made a Zika DNA vaccine. We went into these transchromosomal cows that we've been working with a company called SAB Bio. We can talk about that to make human antibodies. So we were kind of um, testing out our ability to respond quickly to something like Zika. Um, and then it seems like there were some others in there, but then yeah, COVID hits. We were kind of looking at this disease starting to emerge, in China, and we tried to move fast to kind of get a DNA vaccine up and running because what we do, we use the same plasmid that's a DNA vaccine is also what we use to make pseudovirions, go into rabbits quickly to make polyclonal antibody to use as a reagent. So we were doing all those things. And because we had been working with hamsters and um, we as soon as we got the virus, we decided to try and um, see if we could use the Syrian hamsters as a model. Now, there had already been reports out of Hong Kong that the Syrian hamster looked like it was probably going to be a good model, a weight loss model. And so we tried to take it a step further, and we not only went with wild type hamsters, which did get the weight loss and did get lung pathology, but we also immunosuppressed hamsters using cyclophosphamide and dexamethasone, which we've been doing for other viruses. And there you got much more dramatic and kind of extended disease. And so your endpoints are kind of more dramatic. And actually, our DNA vaccine, protected. We were working with some other partners that had some really early on monoclonal antibodies that also protected. And then Joe Golden in my lab, he was able to get his hands on the ACE2, the human ACE2 mice from Jackson Labs. And yes, the virus caused lethal disease in those animals, dramatic lung pathology. And so I think Michael Diamond right around the same time did the same thing. And that model was used a lot for medical countermeasure testing. We had the hamsters up and running. Other groups like NIH, which has an integrated research facility, they call it, which is also on Fort Detrick, where we are. And so the Operation Warp Speed folks kind of wanted to test a large number of different monoclonal antibodies and polyclonal antibodies that were moving into EUA status. They wanted to compare those head to head in an animal model. And so the IRF and USAMRIID kind of worked together to standardize that model and test all of these antibodies in parallel at our two different sites in the same kind of assays. How these antibodies were forming. So in a way, it was kind of like what we had done with the Ebola vaccine, where we had good visibility across a lot of different candidate medical countermeasures. But then, the variants, we got caught up in the variants and trying to keep up with that kind of a running battle. And then the rest of the world kind of really got geared up and became much more efficient at it than us. And so we kind of bowed out of the of that business.
Right. No, it's interesting. When you talk about the theme of, okay, we have these countermeasures, which you're very effective at creating for these various exotic diseases of military importance. And as you're pointing out, it's one thing to develop them, but then it's another thing to properly deliver them and then obviously scale them up. I was thinking, and you were talking about Ebola before, I think we all forget that the ZMapp, monoclonal antibodies, they were, USAMRIID is involved in those, produced in tobacco plants. We constantly have to think about sort of novel production vehicles that we can scale up quickly, that can produce a lot of product. And at the same time, thinking about delivery systems, you've been very active on thinking about, okay, what are some of the alternative, maybe better ways to deliver some of these novel products? You spoke about the Mrna monkeypox vaccine before, and I know you recently did a paper on sort of looking at, intranasal, interrectal, cutaneous inoculations. You have a lot of work on intramuscular electroporation technology. I was wondering if you could take a little time just to talk about sort of what tools, what of these technologies, whether they're on the production side or the delivery side, that you're most excited about in terms about where some of these products should be going as we look at the second and third generations of our need for them.
Yeah. Well, that's a kind of a nice thing about working at USAMRIID is we're soft funded. Actually, the research here, we're on soft funding. So we apply to grants from the DoD, from NIH, from all of those kind of funding sources. But we can also work with industry through cooperative research agreements, and which is really nice because we can work with multiple different companies kind of trying to tackle the same problem.
Yep.
And we can contribute what we can. They contribute what they can. And hopefully out of these relationships, we not only get proof of concept, but hopefully take it further, which is really the hard part. Yeah, so for the DNA vaccines, I mean, it's interesting. DNA, if you get it in, if you can slam it into the cells or get it into the cells, it basically produces messenger RNA, and off you go to produce the immunogen, and your immune system recognizes it and goes. If in our hands, if you take messenger RNA and do that, you can't get the messenger RNA in there. It's because we were trying that back before LNP formulation kind of came on the scene. We were working with companies that were looking at kind of naked messenger RNA. It doesn't really work. Naked DNA actually does work. But the lipid nanoparticle formulation kind of breakthrough for messenger RNA, just, that's what, in my opinion, really allowed those vaccines to really take off, because with very small amounts of messenger RNA, as long as it's LNP formulated, it'll get in the cells and you'll get your response. Back to the DNA. So the DNA, originally we and others were using gene gun technology, where you coat a small amount of DNA on gold beads, and then you ballistically, using gene gun, you put those into the epidermis, the dermis of the skin, and it's very immunogenic, but there's a lot of downsides to it. The basically looking at product stability, potency, all of those kinds of things was really a major headache. It was very difficult because when we finally got up to a clinical grade gene gun system, it was with Pfizer. It's called, I think, ND10 devices. But so they had a helium canister, and then they had powdered gold with DNA on it. And when you discharged it, it would go into the skin, and you'd get a response. And we went up, we did a phase 1 with our got antibodies in humans. It's pretty exciting. The downstream kind of actually making that a commercial product, it was a bridge too far. It was too difficult. And so we went with a different delivery technology, which was intramuscular electroporation. There's a few companies that we worked with doing that. That, again, we get neutralizing antibodies in animals and humans. It looks pretty good, but again, it requires quite a bit of DNA, at least 100 times more DNA than we were using in the gene gun. And there were other drawbacks to electroporation. There's skin electroporation, there's muscle electroporation. It's close to being viable, and it probably is a viable technology, depending on your indication for certain cancers, for other things. But the things that we're working with, it looked like it might be too difficult to get an industrial partner that could really make it economically feasible.
Mm,
So right now, what we're doing is we're looking at jet injection of DNA. And again,
got
this
it.
is all naked DNA. There's no adjuvant involved. It's just DNA. So jet injection, we're working with a company that has a intradermal jet injection. So this is needle-free jet injection or an intramuscular needle-free jet injection. So it's just the liquid that comes right out of a vaccine vial into these disposable syringes, and it's basically delivered at kind of high pressure into the tissue, and enough DNA gets into the cells to actually give you a pretty good response. So we're in a couple phase 2 for our Hantavirus vaccines and a phase 1 for an alphavirus vaccine that we're working on. So that's all DNA. Now, any of this DNA, these DNA vaccines could be converted to messenger RNA vaccines, similar to the monkeypox situation. So we will look into that. We're kind of looking at both those areas right now,
Hmm.
both those platforms right now, because you never know platforms evolve. The good thing about being where we are and the government is not a company, and we can look at multiple different platforms and at the same time,
Hmm.
and move forward. So
So, talking about Hunter for 1 second, because I know quite recently you had a paper published on the quadrivalent polyclonal, which was produced, I think, in transgenic cows, and it just got me thinking. Do you guys do any work? We recently profiled Shavonda Jacobs-Young from the USDA, and she was talking about the MBAF and their BSL-4, that they were I think it's in set up now, it's fully operational, but anything interesting going on in terms of collaboration between USAMRIID and groups like USDA on sort of the agri-terrorism, agri-domain? Because clearly that's something we don't always think about, but we need to keep our cows and chickens and pigs and so forth also protected. It just reminded me when I saw the transgenic cow work, I got thinking about that as well.
yeah, not really. We don't do any kind of agricultural threats unless they really cross over into a human threat. Now, I mean,
Right,
of course, if needed, we can do whatever we need to do, but that is not really our area. That's kind of um, that's the NBAF and with Plum Island.
got it.
That's their area, but they'll talk a lot about One Health, about how a lot of these things they're a problem in animals, and it turns out they're also a problem in humans. So
Right,
anywhere where there's that kind of overlap, we could work with them.
got it,
Uh,
got it.
but the transchromosomal bovines, that's a company, SAB Bio, um, pretty amazing animals that are able to make fully human polyclonal antibody.
That's
And
cool.
so we were able to take our DNA vaccines and we soup them up a little bit with LNP, actually. So we slam those DNA vaccines into the cows, supercharged with the LNP, and we can get a really good neutralizing response. Um, and we actually that quadrivalent anti-hantavirus product funded by NIH, we were able to go up through safety testing, GLP tox safety
Sure.
testing, human tissue binding safety. But then we get to the point where, which is another problem I run into a lot, a market. Um, a lot of the things that we work with are kind of unique to the military or maybe travelers,
Hmm.
or maybe they're a problem in a certain country. And so to try and figure out the market, how to actually get enough interest in an industrial partner to actually make a product, take it the distance, and be able to sustain that has been a challenge, a really difficult challenge for hantavirus vaccines and others. Now, the way some of these problems have been overcome is things like BioShield. So
Right.
for certain um, disease indications, the government has created a market to stockpile medical countermeasures for things that could be biodefense threats. But for things that aren't on kind of that list, um, that the military is still interested in a medical countermeasure for, the challenge is trying to figure out how to partner and fund that.
Got it.
And so things like the quadrivalent hantavirus polyclonal antibody, uh, we need to find a market or we
Sure.
can't move forward. So it's kind of the valley of death we live in.
Right.
Yeah.
Right. Well, it's cool. Cool. I mean, I think the cool part of the story, and again, which maybe a lot of people aren't aware of, is sort of the how far we've come with some of these novel bioreactors, like the transgenic cow, and in the case of ZMAP, the tobacco system. So, I sort of watched the last couple of decades of sort of this molecular farming space sort of evolve, and it's nice to see things coming out of it finally. So that's really cool. Um, Joe, I want to ask you about one other thing. I mean, I mentioned in your bio that amongst the all the nasty stuff that you work with, there was a class of viruses called Mrna viruses, and I noticed a really recent paper that you did on two of these. One called the Junin virus, which is a sort of an Argentinian hemorrhagic
Unine,
fever.
yep.
Yeah, Unin and Machupo, a Bolivian sort of version, and they both kind of sort of their zoonotic source is this kind of cute Calomyes musculinus and Calomyes colossus, this little Vesper mouse. Kind of a cute species, but nonetheless a source of some pretty nasty zoonotic stuff. We talked about One Health. You just mentioned One Health before, in the ag sense, but we talked about this theme with various folks on the show the last couple of years, and there's different strategies out there on, only looking at the stuff in the hot spots versus sort of looking at everything and trying to do a nice swath of all the viruses out there that potentially could spill over one day. I mean, again, can you just talk about sort of the One Health philosophy that you maybe guides you at USAMRIID? Because clearly, there's I guess it's generally accepted. There's a lot of nasty stuff still out there that we don't know about. And whether it's cute little mice or nasty looking rats or whatever the case may be, where are we in terms of sort of a One Health strategy and what we should be doing somewhere between you know, looking in the hot zones versus looking at everything, or what's
Yeah.
your philosophy there?
Well, I think that the NIH is championing kind of a strategy that we've kind of innately been doing, which is um, they call it it's not priority pathogen, but it's prototype pathogens for each family.
Got it.
So arenaviruses, the arenaviridae is a family similar to the Hantavirus. So there's a lot of different families of viruses, but not an infinite number of families.
Got it.
And the idea is that within each of these families, to pick at least one, kind of work out a strategy, like say a vaccine strategy, maybe a small molecule therapeutic strategy, maybe an antibody-based therapy. Try and work out something for one prototype pathogen. And then the idea is if another one in that family kind of pops up, you'll have the kind of a roadmap of how to proceed. And that's kind of what happened with SARS because a bunch of work had been done on SARS-1. And
Right.
so when SARS-2 kind of popped up, kind of they were ahead of the game a little bit. So we've been kind of doing this for a while where we're trying to use our DNA vaccine technology to identify the protective immunogens from each one of these families and at least one virus from each of these families to see what will work and what won't work. For example, with Zika, what we found is while we could make a DNA vaccine that could elicit neutralizing antibodies, we couldn't make pseudovirions in
Mm,
the way that we normally did. So that didn't work for a flavivirus. So Zika's in the flavivirus. So
got
that's
it.
the kind of thing that we found out. And for other families, we found other things like little glitches, little issue you have to overcome. And so it's good to do that with at least one virus for each of these families ahead
Hmm.
of time, so that if another virus in that family kind of turns out to be a problem, you're you're not just completely blind. You kind of have an idea of where to start. So the arenaviruses, we've been interested in those for a long time. Well, actually, Hunin was a biological weapons threat at one time.
Mm,
So, and actually, USAMRIID came up with a vaccine way back,
yeah.
long before my time, called Candid 1 against Hunin, but didn't really have again, the industrial, the market to actually take that further. But actually, Argentina did. And
Hmm.
so Argentina actually took Candid 1 and basically made a vaccine and vaccinated, people at risk in Argentina using Candid 1. So that was, an attenuated virus. Strategy kind of back in the 80s was passage of virus over and over again until it's kind of weakened,
Hmm.
test that for safety in humans, and then you have your vaccine. And MVA is actually similar strategy. MVA is a pox virus, vaccinia passage many times in avian cells till such point that it was weakened. Um, but like Hunin's another example. But the problem with something like Hunin is that's a select agent virus, Hunin virus, and Candid 1. When you go in there and look at the genes,
Hmm.
there's not that many differences between Candid 1 and the wild type virus.
Got it.
So, there's the issue of reversion for all attenuated virus vaccines that don't have kind of the appropriate way that they're weakened, they can revert. Now, there are ways to make them unrevertible, but that requires kind of modern molecular biology to do that, and it can be done. But anyway, so that's Hunin. All these South America, those are all South American arenaviruses: Hunin, Machupo, Guanarido, carried by rodents. And so they could be a problem to the military operating on the ground, sleeping on the ground in endemic areas. So
Got it.
it is something to kind of be concerned about, same as hantaviruses. Um, Other arenaviruses that are actually a bigger problem right now are things like Lassa, kind of old
Sure.
world arenaviruses, which cause a lot of cases in Africa. So there's some vaccines now that are in clinical trials in Africa to try and tackle Lassa.
Hmm.
Um, but yeah, there's some families of viruses that do have some members in them that um, still need to be tackled. Still need to kind of roadmap the best way forward on those. So
Got it. Got it. Jay, what else is, I mean, we've touched on a lot of topics, but what else is coming up that we should know about in terms of you, your team, USAMRIID? I mean, I've seen your name in the press a lot the last several weeks in terms of the monkeypox vaccine, but anything else that we should know about that's going on, that's public facing, and then at the same time, as we get to the end of 2024 and 25, any talks, conferences, other public-facing initiatives that we should know about where we could listen to you, run into you, meet you possibly, and then anything else you want to talk about, please take the floor.
Well, there's a lot of big initiatives funded by kind of big US government, let's say, like NIH has a big U19 project or program called Revamp
Okay.
that play a small role in, but to get it addresses kind of what we were just talking about. This Revamp project is trying to look at prototype pathogens for several virus families. So these are really big consortiums kind of around the country of academic um, industry and government labs that are really tackling vaccines and also antibody-based medical countermeasures against these. So I think that within the next, I don't know, 3 to 5 years, we will have really we will have saturated the identification of targets, kind of vaccine targets, protective antibody targets, and we will be at the point where we're looking at ways to combine targets,
Mm.
increase potency, try and get to single vaccination type vaccines, those kinds of things. But there's such a big effort that's going to be going into really identifying targets and mechanisms and that kind of thing that there's going to be quite a bit of movement there in the next few years, because it's not only the Revamp project. There are other similar programs out there that are looking at multiple vaccine platforms in parallel, using the same targets. There's going to be a lot of molecular vaccine and monoclonal antibody work in the next few years against viruses.
Hmm.
So, I don't want to get into that. Um,
We'll do a
what
follow-up
else?
show. No, it's clearly there's going to be a lot happening, and it's interesting, how we started our conversation off sort of the beginning of your career, and thinking about sort of some viruses that were almost good, had these oncolytic properties, and move forward into the realm of the really nasty stuff, and yeah, it's going to be really interesting to see how the whole space evolves on both fronts, because both of them are quite exciting. Clearly, at USAMRIID, you're focusing on protecting us from the worst of the worst. But it's a really awesome story, Jay, and I really appreciate you spending time with us. Letting us hear about what you've been up to, your journey, and just really wishing you and the team the best as you continue to do this work. Again, for everybody that is going to be joining us and listening on the various podcasts, or who will be watching on the YouTube channel. Again, you've been spending time with Dr. Jay Hooper, Chief Molecular Virology Branch at the United States Army Medical Research Institute of Infectious Diseases, otherwise known as USAMRIID, doing really amazing work to discover these novel medical countermeasures for these exotic viral diseases. Jay, again, thank you for taking the time out of your schedule to talk to us for a little while. Thank you for everything you do there at the USAMRIID to keep us all safe. And as we like to say on our show, thanks for helping to create a better tomorrow via the type of science and technology you're developing. Really an awesome story.
Yeah, thanks for having me. I should quickly mention any opinions I might have stated are my own and not the DoD's. Yeah, thanks for having me. It's a cool podcast. Trying to look at the positive things that people are doing in the world, and thanks for having me.
Awesome having you. We'll definitely do a follow-up. Be well.
All righty.